Thank you so much, Alyssa, all of the organizers. This is an incredible opportunity to be able to speak to you all about really, so many, well, I say one very important topic, but really so many different important topics rolled into this. I also want to say a big thank you to Hillary for the really enlightening talk. And I think about carbon and climate change too, but what I hadn't thought about was that hope is not what we need, and it's courage that we need. And I really think that resonated, and I'm going to hopefully be a little courageous today in showing you. I prepared this talk thinking that I would talk to you about ecology. And then as I spoke to the organizers about this and began to realize how many different facets of the ocean were interesting to people, I thought I would share a more personal story interwoven with some ecology, and that takes a little bit of bravery to share because I'm going to take you to places I never thought I'd go. So today we're going to discuss facts, fury, and the future of the oceans in 3 short examples. And I wanted to thank Chris for starting here with these iconic shots from space. I think we all forget how special these pictures are, right? How hard won they are and how fragile our planet is. And then when we look at these images of this blue marble floating in space, our vulnerability really comes into sharp focus. So that vulnerability is multifaceted. There are multiple different ways in which our world and our oceans, which are of course 71% of our planet, are suffering right now. Overfishing is a big part of the conversation, as Hillary just mentioned, climate change, including warming, but of course acidification, and of course sea level rise, as Hillary mentioned, right, are all part of that equation. And then there's an increasing amount of marine debris, which is entering our oceans at an alarming rate. I didn't go into ocean science to study any of these things. I became an ecologist because I wanted to study life. Thriving, natural interactions, competition, predation, try to understand how the natural world works. But my work has been interrupted. And the work of almost everybody I know has been interrupted by these and other major global changes in the ocean because we can't ignore them. They show up in our samples, they show up in our time signatures, and we can't study ecology outside of the equation of ourselves. So, for me, all paths lead to the ocean from a research perspective. But today, I'm going to talk to you about the fact that literally all paths lead to the ocean. And they do this through a multitude of ways. But one of the biggest, we have many, many issues in our anthropogenic oceans. But one of them, which is something that we just can't ignore anymore, is marine debris. And marine debris doesn't, of course, start in the oceans, right? It starts, I don't know if you can see my cursor, but this big green bar starts on land with the global production of 270 million tons of plastic per year, of which about 8 million tons annually end up in the ocean. And actually very little of that ends up on the surface waters. The vast majority of it ends up immersed in the ocean. And this is an 8 million ton annual contribution to what ends up in the ocean, which has no way out. So some plastic facts just to make you furious. This is actually taken from Waste Reduction Week in Canada, but so just a couple things which apply to us all, no matter where we are in the world. I love this map that Alyssa showed of everybody from all over the world coming today. But roughly 1 million plastic bottles are bought around the world every minute. 8 million metric tons of plastic enters the Earth's ocean annually, like I showed you. 40% of that is generated through packaging, which is something individual packaging has become. There are many environmental potential benefits from COVID, but that might be something where we see a detriment. And of course, very little of this has been recycled. And while there's 8 million metric tons entering the oceans annually, the order of magnitude jump to 8 billion metric tons generated across the world since the 1950s is a terrifying number. So essentially, we're swimming in a mess of our own making. And today I'm going to tell you about three interruptions to my business as usual research life, in this case in the form of surprise plastics, hidden problems and secret corals, and lurking in the abyss. So first, surprise plastics. And for this, I have to give some credit to my 2 fantastic students, Haley Goss and Jacob Jaskill, who, this is just a fantastic picture of them here in Boston, but we went to Belize, a place where I've been working for about 20 years, to study something completely different. We were working on an offshore atoll of Belize, and I'm really interested in parrotfish predation on corals, and parrotfish actually eat a whole bunch of other things. In fact, most of the time they don't eat corals at all. They eat seagrass and algae. And so we were looking at the dynamics of how parrotfish consume seagrasses. And we were doing this really careful experiment. You can see up in top in panel B here, you can see these beautiful pieces of seagrass, and those cute little cutouts are parrotfish bites. And we were quantifying the epibionts, right? The organisms that live on top of the seagrass to see if parrotfish discriminated against eating those extra organisms, those epibionts, or in fact preferred them. And Haley and Jacob were doing this very careful job of characterizing the epibiont structure under the microscope. Here we are, interested in ecology, interested in ecosystems, interested in predation and consumption events. And all of a sudden, Haley says, what's this? And she points out a bunch of plastic under the microscope on the seagrass blade. And my first thought was to dismiss it entirely. I said, no, 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 no. We are shedding plastics all the time, microplastics, right? 5 millimeters or smaller pieces of plastic. I am sure that this is contamination from us. And as we looked closer, it became clear that despite us desperately trying to hope that there wasn't plastic on seagrass, we were wrong. You can see that these microplastic particles are embedded within the epibiont structure. They are literally glued to the surface of the seagrass by other organisms themselves, which means that this is not derived from the clothing that I'm wearing as I'm looking over the microscope, but this is derived from macroplastics in the ocean, which could come from clothing or could come from ropes or bottles or any other, or plastic bags, which are breaking down, floating through the ocean and ending up on seagrass blades. It turns out that the more epibionts you have on a seagrass blade, the more microplastics attach to this. And it also turns out that the more epibionts and the more microplastics on a blade of grass, the more a fish is likely to eat them. Now, it's no surprise that that plastics are bioaccumulating through food chains. But what's surprising is that they're bioaccumulating through herbivorous food chains. So for a long time, or for a while, we thought herbivores might be the exception to the rule. They're not eating organisms, which are then eating other organisms, which are then eating other organisms, which are eating plankton, which are eating microplastics. So perhaps they are immune from this bioaccumulative pathway of microplastic accumulation. And in fact, if you look at microplastic accumulation food webs or food chains, indeed, herbivorous pathways were for the most part left out. The problem with microplastics accumulating on seagrasses and algae is that this means that herbivores are not left out of the pathway. And now there's virtually no pathway, no stone left unturned or no pathway left uneaten for whereby microplastics can't accumulate in the oceans and up the food chain. In essence, this cartoon says it all. Can I have a plastic bag, please? It's in the fish, sir. I'm sorry to say. So back to my normal life for a moment. I really don't study microplastics very often. I study corals, and I study corallivery and things that eat corals. And I work on a lot of different coral-related issues. And one of the corals that I work on is a really cool hidden coral reef here in New England. Yes, that's right. I said a hidden coral reef here in New England. Below the surface of these waters actually lies a single species of calcareous scleractinian coral. This is it. It's a Strangia poculata. This is at the northern end of its range, which you can see here articulated in red. We've been studying this for years. It's a really interesting model coral to look at trophic dynamics, to look at the influence of various aspects of global change on scleractinian calcifying corals. That's not tropical, right? So we have this mouse model for tropical corals that we can use right here in my own backyard. And as we were starting to work on this coral, same story. We got interrupted by finding plastics in the midst of our experiments. So we looked to see if there were truly microplastic accumulations in wild corals at alarming rates. And we looked to see what those plastics looked like. So what you're looking at here, this glob of brown tissue, is actually a coral that has been fixed, decalcified, and dissected. And what you're looking at are the bright microplastic fibers in this top section and this small microplastic bead on the bottom. And it turns out that corals will eat beads, fibers, there's some miscellaneous chips and things like that, right, with other shapes that will break down. But the average number of microplastics that we found in these coastal corals per polyp, right? These colonial animals, if you take a look over here, each one of those little circles is a polyp, was 112 pieces of plastic per polyp. So we put our other experiments aside for a moment, and because there hadn't been work done yet on microplastic accumulation in corals, and looked to see if we could replicate this in the lab. We could feed these corals. You can see the blue fluorescent bead inside the gastrovascular cavity, and hopefully this movie will work. If we feed corals microplastic beads, they readily vacuum them up. Delicious, except not. So we did some experiments. We realized that microplastic beads in brine shrimp eggs could be roughly the same size, same texture, and we could do some experiments looking for a coral preference, hoping, of course, that they would prefer brine shrimp eggs, which, although it's not something they regularly encounter in the wild, of course, have nutritional value and are similar to things they might encounter in the wild. And this kind of experiment is what we did, and it's kind of terrifying because in every single case, whenever we gave an equal number of brine shrimp eggs and microplastic beads to corals, 100% of the time, they consumed more plastics than brine shrimp eggs. Fast forward a step further, we realized that these microplastics they can be eaten. Sometimes if they're beads, they can be spit back out. We were actually watching ingestion, egestion dynamics. So we were wondering if they get eaten and taken in and spit back out, are they actually vectoring microbes from polyp to polyp or coral to coral or population to population? So we coated these microplastic beads with GFP tagged E. coli, fed them to corals, and found that for control polyps up here on the top, we could feed a polyp, we could feed beads that did not have this tagged E. coli, and they were fine. But if you fed them GFP tagged E. coli, and you looked at the focal polyp, within 28 days, there was mortality, and there was mortality in the neighboring polyps as well. So it looks like these beads can be vectors for potential putative disease. And so microplastics are not just a problem from the plastics itself, the ingestion potential, but also the hitchhikers that they carry with them. And if you take a look at this, you can see the blue bead, the green is the E. coli coated the bead, and when you see them merged, you can see them inside the coral polyp. All right, so like I told you, I'm not generally a plastics researcher, so back to my normal world, I often, again, think about corals, and I'm going to take you to my last story, which actually does not take place in this part of the world. So I'm not thinking about the Atlantic. I'm going to shift your focus to the Pacific for two reasons. One, of course, is to let you also see how big the Pacific is, right? And the United States government, as part of a 3-year exploration tour, up in this top corner, you'll be able to see the ship tracks over a 3-year tour of the US explorer ship, Okeanos Explorer, which was going to do deep sea exploration. I mean, deep sea, lower than 200 meters, down to 6,000 meters, all over Pacific remote islands, marine national monuments, marine protected areas, and this pink protected area is the Phoenix Islands in Kiribati, and this purple spot is Papahanaumokuakea. And while this US government ship was out exploring the oceans and looking at beautiful and long-lived deep sea corals that live for 1,000 years, 2,000 years, deep sea sponges, which are just incredibly beautiful, colorful, look Zeus-like, and have all kinds of interesting ecological roles that we're just beginning to understand. Of course, when you think about the deep sea, we sometimes think about the unique creatures. Sometimes people think about shipwrecks. Sometimes people think about Amelia Earhart's plane crashing. And unfortunately, those anthropogenic pieces are part of the story, right? Even on these deep sea cruises where you're going up these underwater mountain chains with just a relatively large ROV, but just these lights, these tiny little trails that you can only see what's lighting up in front of you because it's so dark that no sunlight shines. You see these incredible organisms, and you also see us on the bottom of the ocean. So not in high numbers, I'm happy to report. So in the far-flung remote ocean, we do see marine debris, but less so than we do in our coastal oceans. But my point is that no matter where we are, we can't escape ourselves. And in fact, a study was just published this week that showed that 14 million tons of microplastic was accumulating on the seafloor, even in our global oceans. And that's even in the remote deep sea. The future, the good news. There are credible paths to significantly reducing plastics in the ocean. but they require multiple solutions. Oops, sorry. They require multiple things to happen at once. A business as usual scenario is going to increase our plastic consumption, use, and deposition in the oceans. But if we can reduce, substitute, recycle, dispose of, and revert mismanagement to proper management, then a lot of exciting things can happen. And this was a paper just published by some colleagues at Pew in Science very recently, just 2 months ago. And I think it's a really exciting opportunity for us all to assess how we can put multiple solutions together so that we can think about how to solve this problem effectively and quickly. And their models showed that we can actually achieve an 80% reduction by 2040 if we put multiple management strategies into place. Just to wrap up, like I told you before, I didn't become an ecologist to study plastic. We have to study it sometimes because it's in our way, and to ignore what we see in front of us is to not do our science and our scientific training justice. But I love this quote. The greatest wonder of the sea is that it is still alive, despite all that we've thrown at it so far. And that gives me hope, or as Hillary pointed out, it should give us courage to do what needs to be done. And so the vast majority of my time I spend thinking about this tiny or big, depending on how you want to look at it, protected area in the Central Pacific, owned by the Republic of Kiribati, who has had the courage to protect one of the most important places of the ocean. This is a country, a least developed country, that has put together a marine protected area the size of California and has made it no take, protecting it against all kinds of ocean problems. However, even these lines can't keep out climate change. This is a spot that was beautiful in the year 2002, following several marine heat waves, had almost 100% coral loss. But what places like the Phoenix Islands can show us is that corals can recover when they're not dealing with everything at the same time. This place, this California-sized box in the Central Pacific, has deep sea, it has ocean seamounts, it has islands, it has a ton of open ocean. There's a lot of stories to tell here, but importantly, it's protected. It's protected against overfishing. So these are the fishing days before it was closed, and now here's how much fishing effort is happening after it's closed, essentially none. It's been very effective. There are very few people. There's no permanent population of people living there and constantly throwing debris into the oceans. And even though it's very sensitive to El Nino dynamics and climate change, it's a chance for us to study nature in the best of our ability in this modern world. It's a place where there are so few people that we can ask the tough questions about how much can our oceans handle when we're not right there adding more stuff into it. And so all paths lead to the oceans, even remote oceans, even in the deepest of seas and in the most remote of areas, we see evidence of ourselves. And this is really obvious when you think about the way we live our lives and the fact that we all live in watersheds. You know, Hillary ended on a map of the United States, and I will too. You know, when you look at the United States from a watershed perspective, you know, I often look at the Mississippi River watershed and I think about, my God, if I was an oyster living here, what would I experience? And even if you're not living in a place, I saw that beautiful map of people all around the globe, you know, listening today, wherever you are in the world, what you do in your backyard, in your daily life, in the municipality that you belong to, and in the votes that you make and the actions that you take contributes to the ocean. And so with that, thank you. And I'd be happy to take some questions.
Thanks, Randi. Fantastic. Indeed, the oceans are pretty sick, but there's still lots of amazing life going on there, and lots of reasons why we can aim to improve things in the future. As you say, there are paths forward that are doable, right? It's taken us a long time to appreciate just how bad all of this plastic has been for the ocean, and I think now we pretty much appreciate that. And of course, it's ugly to look at. It's very sad to see all of that plastic in these beautiful places that you have shown us. But how much is plastic debris actually changing marine life, would you say? How much is it changing marine ecosystems? You know, it's something new in the ecosystem that has only really started to appear like after the Second World War. Right. And do we know how much, whether it's changing evolving, leading to evolution of life somehow in the ocean?
I love that question, and it's a really important one. And I would say that I think the science is, we're at the beginning of answering that question rather at the end. And so many colleagues of mine throughout the world do study plastic pollution full time. These three stories are ones that, as I said, have sort of interrupted our thing, some of our normal work or things we've come across. But what I would say is that there are studies that show that plastics can interrupt food acquisition and digestion in a number of organisms, including plankton, zooplankton, which is very important because they sit at the base of the food web. And if we have major loss of zooplankton due to either starvation or whether it's, the mechanism of mortality, it could either be starvation or it could be disease vectoring, or it could be, a number of, or chemical leaching, right? There's a number of different things that could contribute to this. But a loss of a major part of our basal part of the food web is really problematic. Some of the other issues may be more subtle, meaning that they may not be immediate mortality, but they may have sublethal effects. And in fact, I would venture a guess that that's where we'll see the majority of the interactions and a lot of the impacts of these plastics. Sublethal effects that we don't fully understand for a while to come. But the reality is that we are eating plastics. We are drinking plastics as well. And we don't know the impact. We don't fully understand, I don't think, the impact on ourselves or on marine life either. And the chemical leaching is one piece of this. There's also the structure of the plastic itself. Imagine 110 pieces of microplastic fiber wedged into the gastrovascular cavity of that coral I showed you. Of course, it's going to affect the structural matrix of the way that organism is living its life. And in addition, of course, the microbial vectoring, I think, has the potential to, if it can be ingested and egested, we're in a global pandemic. I think we're all thinking about infection in different ways than we ever have before. And I think paying attention to how that might happen in the ocean, pandemic aside, is I think, one of the areas that's ripe for more investigation.
Yeah, I would just like to ask one question, Randi, because I know that some of our participants are eager to have lunch. But I just want to say that that was just absolutely fantastic, and the connections to Hillary's talk are great. And I think everybody will see the connections to this afternoon later. So I'm just going to ask one sort of physics/biology/chemistry question, which is, we heard Hillary talk about the difference between basically the heat capacity of air and of water. And what that translates to is that it takes a very long time to change the temperature of water compared to change the temperature of air. And so I'm curious, with the plastics, and the coral reefs, you showed the coral reefs recovering over some period of time. So are people thinking about what the interplay is between, say, the temperature of the ocean, the acidity of the ocean, and then the amount of plastic? Like, I assume that the timescales involved in changing things, it's all interdependent, the physics, the chemistry, and the biology. So that's maybe a question for our discussion later, but I guess now you can just go, yes, people are thinking about that, or tell me how they're thinking or they're not.
I'll keep it quick. I don't want to keep people from lunch, but I guess I'll say that the thing to avoid is death by 1,000 cuts. Right. So all of these things are happening simultaneously. I don't think we can talk about climate change by itself or plastic pollution by itself without also putting this all in the context of human demographic change. Right. We have a huge global population of people. A lot of the things that we do wouldn't be so bad if we had, I don't know, a third of us or a quarter of us on the planet. Right. Which is where we were, a mere 100 years ago. And so thinking about all of these things simultaneously, I think, is a growing area of interest. I think it's really hard to put everything together. And so some of the most effective work is coming from modelers and meta-analyses that I think are able to put the relative context of each one of these things together across multiple ecosystems and organisms. But we can have a much bigger conversation about this afterwards. And I'm hoping we do, because it's a bigger conversation.
Perfect. I'm so pleased with the range of speakers that we've chosen. And so I know what people are going to speak about after lunch, and just come back, everybody. So thank you very much for now. And I'll sign off for right now. Thank you.